Okay, so let's get started. Let's see, I think this is what I use. Oh, okay, yeah. So I'll go over this very quickly. How many people don't know what Midas is? Okay, so I'll go over the introduction very, very quickly. We're a university-wide organization supporting data science and AI, both the methodology development and also the application across research domains for scientific discovery and also for societal impact. So we enable a lot of research. We offer lots of training for faculty, staff, postdoc students. We build a lot of collaboration across universities, industry, and government. So this Data and AI in Society lecture series has been running for more than a year now, and this is our dedicated event for non-technical and also for the entire community, U of M and also external people. And so the reason that we started this series is really because AI and data are transforming society and transforming science in such profound ways. We really need to collaborate across organizations, across with community to really figure out how we basically, well, let me put it plainly, how we deal with AI. So this is something that we really really want to promote here at Midas. And so we have two more lectures coming up in this series. In March, we have Chairman Budek talking about how to use AI to support civil discourse and the exchange of ideas. And then in April, we will talk about AI, how AI is being used to support Detroit, especially population and census data. At the end of March, we will have the AI in Research Symposium. 3 pretty awesome speakers covering physical science, biology, and also AI and music. And we also will have a large number of University of Michigan researchers presenting, we call them AI journeys, and these are about how, I always say this is about how the sausage is made. And so they will talk about how they choose research questions and AI methods and overcome obstacles. So we also have one more generative AI tutorial coming up on March 19th. It's about building agents for research. Housekeeping items. I think bathroom is over there across the big space. In the event of fire or evacuation, use these doors to go out of the building. Q&A, wait for a staff member to hand you the mic. Try to keep your questions brief, and also remember that after the lecture, you'll have the time to talk with Dr. Cernak. So Dr. Tim Cernak, he's a faculty member in medicinal chemistry and also the Department of Chemistry. So Tim really is a pioneer in using AI to not only make discovery, but also to vastly accelerate the discovery process. And he's been doing this since before many people knew what AI is.
We didn't call it that.
And so most recently, he started getting into new research directions, such as protecting endangered species using AI. I actually read your story. That research was featured in New York Times. And so when I saw that story, I was like, wow, it's so cool, but it's not surprising to me at all. It came from Tim because he's one of those really creative researchers. So I'm really happy that he's here today to deliver this lecture.
Thank you. Thank you so much, Jing, and for that kind introduction, and to Jing and Ben for this opportunity. And thank you all for coming out on this chilly evening to see this rescheduled event. And let's now try this. It's coming. This computer needs to submit a grant on Monday, and it's very sad right now. But it's going to get through today's talk. It's really my great pleasure to be here to give this talk in this building, and I'm going to make this clearer why quite quickly. This is a great opportunity for me to present this new idea that Jing highlighted. We call it conservation chemistry, and I'm excited to show to you what we're thinking about and how we can use AI, we hope, as one of the tools in this fight. And the reason that it's interesting to me to give this presentation here in this building is because of this paper. So in 1985, Michael Soule was an adjunct faculty of, at the time, the Department of Natural Resources. And here out of this building, launched a really provocative idea. He gave this paper saying, what is conservation biology? And explains this, what he calls a crisis discipline, that we were living in a biodiversity crisis all the way back then, we are today. And what can one do, right? The biological sciences had some tools that could give insight to preventing the extinction of plants and animals. But natural resources, the Department of Natural Resources, for example, or the Department of Fisheries in the government plays a critical role in making that happen. And there was really no avenue by which a biologist should be talking to the Department of Fisheries about the conservation of an animal, and also thinking about the population genetics upon which that could be thought about. And so here's Dr. Soule back in this era. He was a field biologist and really had a specialty in population genetics. And so today's conservationists would say, well, if we want to prevent the extinction of this species, we're going to need a founding population that maybe a zoo would go and care for that founding population enough to get there to have a genetic pool to work with. These were principles that were being explored in the 80s, and today we understand this well. Zoos can prevent the extinction of many creatures, and other conservation biology institutes. It's obviously not just zoos, but it was a very controversial paper because it was described as a crisis discipline, and there was no history at that time of biology was a basic science that was really meant to focus on just the scientific discovery and not so much the action. So he actually faced quite a bit of headwinds in trying to explain this work. Here, this is one of the figures from the paper, and so he explains how cancer biology was well understood. It was an intersection of pharmacology and virology, cell biology, molecular biology. It was easy to explain what a cancer biologist would do in the 1980s. But conservation biology, as I pointed out earlier, it has genetics as a part of it, and population ecology as a part of it, and veterinary medicine as a part of it. But then also, things like forestries and fisheries and public policy are important components of this new field. And so this was, this was launched from this building in 1985. And as I've been trying to learn about this space, I sit in on conservation biology classes taught to our undergraduates, where there's whole textbooks on this field, and we understand what conservation biology is. There will be some upsetting images in my talk, and I apologize for that, but I think we need to talk about some things. So this is a population of elephant seals in Argentina last year that were completely wiped out by avian flu. We lost 98% of elephant, at least the elephant seal pups of that year to avian flu, and we're in this situation where this is happening around our world right now. that there are diseases that are causing massive conservation challenges. And I was really impacted by COVID in my research. I didn't have any health issues per se. I've had COVID 5 times, but it's always been pretty gentle with me. But it really kind of messed me up when that disease hit. I was like, what if this is it for our species? Like when it came out, I was like, as a chemist, I'm a chemist. I'm a medicinal chemist. I've made drugs for a larger part of my career. It's like, what can I do in this space? And I'll try and share more about it. And so we're launching this idea of conservation chemistry. It's been very difficult to break into conservation as a chemist, and I'll talk about some of the reasons why, but we'll talk about some of the ideas that we have within our lab of how we can make an impact in these exotic diseases. Before we do that, though, I need to do my best to get you excited about chemistry. And when I got to organic chemistry, I was like, this is what I want to do for the rest of my life. I want to draw hexagons and look at molecules like this. This is my worldview. When I'm looking at a problem, if I can't look at a molecule associated with the problem, you don't have my attention yet. And so in my PhD, I explored natural products like these ones. I considered ways that we could use synthetic chemistry to stitch them together. With those skills, I enjoyed a career in the pharmaceutical industry. I worked 9 years in big pharma, and I launched a biotech in La Jolla doing synthetic work. And then today we enjoy this intersection of AI and pharmaceutical research. There are I don't know the level of investment, but I know that the pharmaceutical industry is about a $4 trillion industry at this point, and they are deeply invested in AI. Imagining that we could design molecules, we could make them, and we could test them towards all types of diseases. You can imagine, maybe in my lifetime, I'll get to see treatments for all kinds of diseases that come out at a rapid pace. We did provide treatments for COVID, right? Within 1 year, we had a vaccine. Within 18 months, we had a small molecule medicine. You can take like a pill in this form, and that's not the norm yet, right? There was an existential threat to humanity that accelerated that discovery process, but it's getting faster every day. That AI is accelerating every facet of drug discovery. There is a multi-trillion-dollar industry that is pushing it as fast as they can to make this, even faster. And so I do imagine a future where we have a big red button that you push and it goes design, make, test through the drug discovery process and treats a new disease that we don't yet currently have a treatment for. Like I said, COVID was very impactful to our lab, and I just want to share one small vignette and then get deep into organic chemistry. I hope that you'll just enjoy the bonds and pictures, and then I'll bring it back to something that will be easy for all to follow. But I hope that you can leave this talk knowing a little bit more about organic chemistry. We were sponsored by a chemical supplier when COVID hit, and they were very concerned that they were about to get an order for billions of doses of a starting material to a drug that they had no idea which one it was going to be. So in like February of 2020, they're like, we know that we're going to get hit with a request for lots of chemicals for some drug. We don't know which drug, we don't know which chemicals are going to go into them. And so they approached us and asked, would it be possible to find backup supply chains to all of the COVID drugs that are being investigated right now? And so they said, we want you to look at these 12 drugs that were undergoing clinical trials in February of 2020. And we want you to find synthetic chemical routes to them that don't use any of the starting materials that are currently used in their commercial production. They gave us 8 weeks to do it. And we found these routes using a form of AI that I'll skip over for tonight's lecture. But the point is that the drug is the yellow dot at the center, And the little spider web of dots going down is the synthetic route to that drug that's used in commercial practice. One of them is in commercial practice and others are used in research practice. But the little orange dots, hopefully you can see these tiny little orange dots at the bottom, these are the starting materials that the chemical supplier would provide that get stitched together to make this drug, baricitinib or favipiravir or umifenavir or bromhexine. These come from chemical building blocks that need to be stitched together to make a chemical bond using synthetic reactions. And my lab is invested in making such reactions and exploring such synthetic routes. Anyways, we used an AI to describe a way to get to the purple and pink dots going up. And critically, those purple and pink dots are not the same as the orange dots. And what that meant is that should the supply of one of these drugs should we need to provide billions of doses of one of these drugs, we provided a backup route to each one of these. Now, these were all the drugs of interest in March of 2020. Of course, the drug that became most interesting was Paxlovid or nirmatrelvir, which is not on here. It wasn't invented yet. But I just want to give you a sense of one of the ways that we've used AI in my lab to solve supply chain issues for disease challenges. We're already deeply invested in using this same strategy for achieving total synthesis of small molecules. And I show one here. This is a molecule called stemoamide that is a natural product we were very interested in. And we used this same tactic to make this molecule in just 3 steps, which was a very proud achievement for our lab. This was the 34th synthesis of this molecule, but the shortest by a mile. And so you don't need to understand the details of these steps other than to say that in 1, 2, 3 steps, we take this pretty simple thing and build it up into this rather complex molecule. And so that was an exciting moment for our lab. And one of the things that informs us and gets us thinking about this design, make, test, how could we accelerate that? We're the makers of the drug industry. We're the ones that focus on how to make the medicine and the reactions that we use inform which molecules we choose to design. And so one of the things we love to do in this lab, we'll get a little bit deeper into chemistry and then I'll pull it all the way back. We'll talk about zoos and conservation. But the multi-trillion-dollar pharmaceutical industry gravitates to just 5 chemical reactions. And the top one is this orange slice of the pie called the amide coupling. If you've ever seen a protein or a peptide, it's got a backbone of these amide bonds. And so you take an acid and an amine and you click them together to make this amide bond. We recognize that was one particular reaction that was highly important. My career in the pharmaceutical industry certainly illuminated to me the power of this reaction. We would use it all the time. But I asked the question, why is that the reaction that we care about? Why is it that we take this amine here and this carboxylic acid and have them unite to make this amide? Now, if you've not seen chemistry in a long time, you can't follow that necessarily, but hopefully if I tell you that this molecule is basic, and this molecule is acidic, and this molecule is neutral, that will be okay. As a drug hunter, one of the things that we typically think about is the properties of our molecules. Are they basic? Are they neutral? Do they have hydrogen bond donors on them? If I'm trying to treat Alzheimer's, I'm trying to get a medicine that will go up into the brain. And the number of hydrogen bond donors on the molecule is going to impact whether it gets into the brain or not. So as a drug hunter, I need to think about these properties. So our idea was, why is it that this multi-trillion-dollar pharmaceutical industry loves this one reaction when in fact there are hundreds of other possibilities? There's even more than hundreds. You can draw them on a slide like this, but there's no way yet to go into the lab and actually make them because we haven't found a catalyst that would make that possible. We do use AI to find catalysts that can make this possible. So far on our journey, we've achieved this reaction, this reaction, this reaction, and this one actually just got published. Sandeep Das is in the lab and is a first author on this publication. We're very excited about that work. So I was always taught when I was in big pharma that, you know, when you want to change the properties of your molecule, you've got something that's targeting the pancreas and you're trying to target the brain. You want to lower the number of hydrogen bond donors. You go to your big box of Lego and you get pieces of Lego that have different properties and you stitch them together. Our question is, what if we only had 2 pieces of Lego? Can we manipulate the properties by connecting them together in different and interesting ways? And so this has been something that our lab has been deeply focused on. It turns out that if you take this very simple amine and this very simple acid, so this is called ethylamine, and this is called propanoic acid. And there's a carbon here, there's a carbon here, there's a nitrogen here, there's an oxygen, a carbon, an oxygen, a carbon, and a carbon. So there's 8 heavy atoms in this entire system. If you throw those atoms in the air, you let them land so that carbon gets 4 bonds, oxygen gets 2 bonds, nitrogen gets 3 bonds. There's 56 million ways that they might come together. One of them is this funny guy here. We have no idea how we'll ever pull that off, but we're going to keep trying. You know, the pharmaceutical industry, this multi-trillion-dollar industry, would use just one of those 56 million opportunities. And yet each one gives us a different set of properties, a different way to think about the molecules that we're making. And we can change the properties of our molecules by changing the way that we stitch together these atoms. So here's a simple amine, a simple acid. We stitched them together to make an amide. This is called an amine. It's a bit more basic than the amide. This one is very aromatic, so it glows bright colors when we shine fluorescent light on it. This is a carboxylic acid, and then if we work this up with copper, we get this really greasy lipophilic thing. And then finally, we had another copper catalyzed reaction that we can make esters. And so all that you need to understand is that we're changing the properties of these molecules. That's what's shown with these little mountainous blue curves. But we're doing it by changing the catalyst, and that's a really different way to think about changing the properties of our molecules. And we did this in partnership with Johnson & Johnson. They've been great supporters of our research. And we used these different catalysts to make these different bond arrangements. Here's an amide, here's an ester, here's a carbon-carbon bond, here's a ketone, and here's an amine. And we stitched them together to take these drug-like molecules and these so-called degraders, but the linking atoms are the ones that are highlighted here, are these different amide, ester, alkane, ketone, or amine groups. And what we see from this is that the way that we link it together can have a profound impact on the potency of these molecules. So these were molecules that Johnson & Johnson was treating using as cancer therapies. And what they see is that based on the linker that we choose, we can get something that is 95 nanomolar, which is pretty good. But if we go to the ester, we're 5 nanomolar, it's 20 times better. And then the amine is completely inactive. We don't see any activity at 11 micromolar concentration. And the little movies I just showed explain a little bit about why we see these differences. But critically, the point I wanted to display here is that by changing the way that we link these 2 pieces together, we can manipulate the function downstream, right? So we're changing the IC50 or the dose, basically. You can imagine that this is a 95 milligram pill versus a 5 milligram pill. When I go to the pharmacy, I tend to gravitate to the smaller pill. So, you know, just being able to change the dose of your drug by changing the reaction that you use to produce it was a new principle that hadn't been looked at before. And so there's 56 million ways that we intend to unite an amine and an acid. So far we've discovered 8. This one says in preparation, but as I mentioned, Dr. Sandeep Das has just published this in Jack's Gold, and we're really excited about that. We've had a good run of developing these reactions using robotics. I won't focus too much on our robotics platforms today, but we do generate lots of data using a liquid handling robot so that we can then apply machine learning models on the data that we generate in hopes of understanding and predicting into the future new chemical reactions. And so I believe this is getting close to my final chemistry slide for a moment. If we use our robotic platform, we can print out 1,500 little droplets like this of little liquids that have future drugs inside of them. This top rectangle is showing us that we were able to make a lot of these different drugs. The purple is bad and the yellow is good. So there's a decent amount of yellow on this plate. That's good. We then dose these into prostate cancer cells at 10 micromolar. And you see that there's a whole lot of purple, just a little bit of yellow. We're okay at killing prostate cancer cells at 10 micromolar concentration. And if we go to a lower concentration, think about having a lower dose if you went to your pharmacist, we kill just a handful of cancer cells, or each dot here represents a different potential anti-cancer therapy that is good at killing prostate cancer cells. But what I aim to exemplify here is that we can navigate drug space very quickly using the technologies that we've been developing for well over a decade now. And you know, you get these kind of rich data outputs. These were all various cancer programs we were working on. This was in partnership with Merck and Company, my old employer. This was in partnership with Relay Therapeutics, and this is based on a natural product that we're very interested in our lab. And so different cancer programs where we're generating lots of data, trying to hone down, use machine learning models or AI models to navigate through this data to find the best compounds that would be least toxic for our patients and most efficacious, so that we can take a small pill rather than a big old horse pill you got to take every couple hours. And so we think about this all the time. How could you design a medicine? How could you make the medicine as a synthetic chemist and then test it in some way? Think about the cells that the cancer cells I just showed there. And so we imagine, you know, that in the future this will continually be accelerating, this design, make, test, design, make, test. I think it's quite reasonable that the pharmaceutical industry will be producing medicines at a much more rapid pace than they are today. And I imagine a future where, you know, a next pandemic hits and we don't have to worry as much as we all had to during COVID, that we would have better pharmaceutical options that could become available more rapidly. And so one of the areas where we've been spending a lot of our time is looking at avian flu. We were very concerned about that from a pandemic perspective. And so one of the interesting things is that today we can look at mutant proteins that are evolving in the wild, and rapidly fold them up using the AI tool AlphaFold, which I just showed here. This won the Nobel Prize in 2024 in chemistry, and so what I showed you just there, this movie, was a mutant of avian flu that we're very interested in, and a candidate molecule is the little yellow guy in the middle, and it's the various energy interactions that it has with these proteins are these squiggly lines that I show here. So Nick Tripolitziotis is joining us today, and he's actually working on making exactly these yellow molecules as we look at avian flu mutants. And so we're very interested or very concerned about avian flu mutations from the perspective of drug discovery and you know, just making sure that we would have pharmaceutical options if anything should happen. You know, you've heard about avian flu in the news. It's impacted the price of eggs. It's impacted our milk facilities. And we're very concerned about a potential spillover event, but thank goodness there hasn't been any significant human-to-human transmission. And so what we do is we would take one of these mutant proteins, append it to a magnetic bead, and then go fishing through one of these libraries of synthetic chemicals that we've made. We can design the molecules using AI. As I showed, we can make the molecules using AI, and we can design the proteins using AI. In the old days, we would have to do X-ray crystallography to get a new protein's chemical structure to show you that gray wiggling blob that I just showed you on the last slide, and understand where we want to have a molecule binding into it. And so we imagine a future where it's going to be easier and easier to make medicines. And we got into it because of these guys. And so this is the California condor. There were 500 of them left on the planet, and 21 of them died to avian flu recently. And that set back the conservation of the species by about 3 decades. And as a drug hunter, I just, you know, I was really moved by this moment. I was like, I can't imagine a potential extinction of a species from a disease, right? I mean, I've dedicated my career to making medicines for disease, And yet there are all these diseases around us that are causing mass conservation challenges, driving the extinction of animals. It's just that, they're condor. So their healthcare isn't great. It's not bad. We know where every single condor on this planet is. Each one has a name. They don't all have radio trackers, but we know where they are. And so luckily, by the time 21 of them had died, the remaining 480 were vaccinated. They were the first creature on the planet. Well, there were black vultures that were vaccinated to trial the vaccine for avian flu, and then the remaining 480 condors were vaccinated, and so they're no longer at risk of extinction from the current strains of avian flu, but they certainly got us thinking about this problem, and this was really what accelerated our research in avian flu, particularly was reports on condor. It was bald eagles and owls right here in the Midwest that were really motivating me, and so it got us thinking deeply about if we have this design, make, test idea, and if it gets faster and faster to make a medicine, what's going to happen when we can make a medicine for every human disease, right? What if that's a future that we all get to see in our lifetime? I think it's not every single human disease, but it's maybe not impossible that you or your child will see a future where most diseases could have a treatment produced for them. But then, as we do live in this biodiversity crisis in this moment, what about a butterfly or a turtle or a tree? What would happen to them if it was a disease that was causing their extinction? Could we make medicine so fast as to engage in that? Now, there's all kinds of really big ethical questions that you probably have on your mind, and I'm going to try to unpack some of them, and I'm excited for conversation after the talk as well. But one of the challenges is that it's been quite difficult for me as a chemist to find my way into this space. It's truly my great pleasure to be standing here in the Dana building to deliver this lecture to you guys today. I mean, the historical significance of this building to conservation is very much not lost on me. And so, conservation biologists would have said, well, if we're going to conserve the panda, we're going to need this many acres of a nature preserve. We're going to need in vitro fertilization techniques at the San Diego Zoo or the Smithsonian Zoo, in many facilities these days are involved in panda breeding. We'll need some regulations to go along with it. And so I think that's really fascinating, but almost every panda, at least all the captive pandas, have been treated with albendazole. And so now you're talking my language, right? Like now I can engage in this conversation because I know about this drug. And so I find that really interesting. I don't know about the pharmacokinetics of this drug in panda, but I think we could definitely answer that type of question. And so it took a while to formulate this, but I want to lay it out for you here. So what is conservation chemistry? We define conservation chemistry as the molecular level study. I would like to look at an organic molecule, but proteins, DNA, I think would fit into this as well. But there needs to be a molecular level insight that is coming from this into one of the 6 drivers of extinction. Now, what are the 6 drivers of extinction? If you sit in Conservation Biology 101, you'll see this list. You sit in many classes in this building, you'll see this list. There are 6 primary drivers of extinction on the planet right now. There are invasive species, disease, habitat loss, pollution, overharvesting, and climate change, which is kind of an umbrella for a lot of things that are driving some of these other possibilities. And now it's rarely the case that just one of these is what's driving the extinction. Oftentimes they synergize with each other. But if you talk to a conservationist, they'll tell you that these are the 6 individual aspects that we can focus on. And so my lab has been focused largely on disease, as I've talked about. We also are focused on invasive species, which, by the way, it's invasive species week. So go kill an invasive species somewhere. Right. So these are some of the creatures or plants and animals that have captured our attention. Many of which we've engaged in research on. Others have inspired models that we've tried to look at this problem. But with each one of these guys, there's a very serious disease challenge that is threatening its existence on this planet. And it's kind of fascinating to think about that. I mean, these are, if we were to say there's a new type of cancer, if we were to go to the hospital and a surgeon said, I just took out this new type of tumor, we've never seen anything like it. We could set the resources of the modern pharmaceutical industry, of cancer researchers here on campus towards this problem, and we'd know exactly what to do. It's the same thing in diseases in all of these species, right? I mean, there's definitely differences. There's a lot of ethical changes that one needs to talk about. Handling an endangered species requires a lot of paperwork. But the disease processes are largely the same. A lot of the biology is quite different. Sea turtles do not have a blood-brain barrier. We have a very powerful blood-brain barrier, and so when I'm treating most patients with human drugs, when I'm treating human patients with human drugs, I don't worry about brain toxicity too much because almost no drugs get into the brain. But for sea turtles, they don't have a powerful blood-brain barrier, so like the biology is a little different. That makes it interesting. Let's talk about sea turtles. So I've dedicated a lot of my career to work in cancer, as I alluded to. And we do imagine a future where we can use these high throughput robotic tools that I described, coupled to AI to make cancer therapies in a rapid way. And so when I saw this picture, it really, really affected me because I had spent so much of my career working on cancer. And I just had no idea that something like this was a problem. It turns out that this is a cancer that is the primary reason sea turtles in America wash up on the beach today. They get it so bad all over their face and between their flippers that they can no longer swim. They can't see where they're going, and they just wash up on the beach. And we work with a sea turtle hospital that collects about 5 animals a week in this condition, And so we went down last year and met one of them. This is Bellatrix, the sea turtle. And you can see she's not in a great way. This is Chun-Yi Tsai. You can see her reflection there. She's a chemistry PhD student in the lab who was studying this problem with me. And Dr. Muhammad Ishfaq is a veterinarian who's used to be on the team. And so we were going down to learn about this problem and try to imagine how we could use our tools, our perspective. How could we find a protein within one of these tumors that could be relevant to this disease? We've got hundreds of cancer drugs in the pharmacy today. So maybe there's some way to tackle this problem using existing tools. Well, we got to witness the surgical removal of these tumors from Bellatrix, the sea turtle. These were all the drugs that she got throughout this procedure. The veterinarians were injecting this one, and this one was inhaled. This is betadine, is just like a brown liquid that they were kind of disinfecting wherever they were making cuts. And I was there as a chemist, just like tell me more, just like drawing the structures of all of these things, trying to learn about this. But not a single one of these is a cancer drug. Right? So Bellatrix, the sea turtle, got all of these drugs to deal with her cancer. Not a single one of these drugs is for cancer. They're pain meds and sedatives and disinfectants. So within our lab, we're very eager to study this problem and navigate through sea turtle cancer cells like the ones I show here to try to find proteins that might be relevant. If we could find a biochemical pathway there, we'd have the tools to match it to to the modern cancer therapies that exist today. And the wiggling proteins that I just showed you on the last slide, again, this is one of the ones that using modern AI, we're able to fold up a protein like that and get its three-dimensional structure and start to do drug discovery. That was a protein from a sea turtle. There has not been a protein crystallized from a sea turtle. That takes a lot of effort to do. But using modern artificial intelligence tools, we can at least get our feet beneath us and get started in this problem because we can start looking at sea turtle proteins in the computer before we could even have experimental access to crystals of them. And so we're hopeful that that we can bring some of the things that we know from modern medicine to this fight. And yeah, let's just enjoy a video of little baby sea turtles for a second. All right, so let's go now to the forests of Michigan, but really the forests of Pennsylvania. I'd like to talk about the state tree of Pennsylvania. This is the hemlock tree. It's my favorite tree. And the reason I love this tree so much is because it's a conifer that can grow in the shade. It's in a very bad way. It's been impacted by an invasive insect called the hemlock woolly adelgid. And so this is the photo from the Great Smoky Mountains. Every tree that you see that is green is not a hemlock tree, and every hemlock tree that you see is dead. And so this insect was introduced into a Japanese garden in 1951 in Virginia. Someone brought in a bonsai tree with an insect on it, and since then it has really had a devastating impact on our hemlock forests, starting here in Virginia, and then, as you can see, working its way. It reached Ann Arbor in 2023, and so we'd been working on this bug for quite a while, and then finally we got to actually see it. I wasn't happy to see it, but it's here now. We are working with the Nichols Arboretum, where we're testing an insecticide that we believe will be a bit more targeted to this bug over there. It implants in the tree, and it creates this little white waxy coating, and we're hoping that we can use that as a potential tool to address this insect. Here's the tree, one of the trees that we've been studying in Saugatuck Dunes. And so you see here the power of this tree. So it's surrounded by a maple forest, right? And you can tell it's winter. All the maple trees have lost their leaves. Yet this tree and a couple of its buddies living in the shade remain evergreen. We have pines and junipers and spruce here on campus. They require full sun. They can't grow in the understory of a forest. And so this is the only tree in Eastern North America that has this property where it can grow as an evergreen tree in the understory of a deciduous forest. And hopefully you can see from this photo the impact that has on the ecosystem. This provides critical habitat for birds migrating through, for deer, and they're really important to the trout fishery, keeping streams and rivers cool. It's just my neighbor built a terrible eyesore of a building in a shady spot, and I only wanted to plant a native plant, so I went to the garden center and said I need a tall conifer that will grow in the shade, and it's got to be native. And he said, well, a hemlock is what you're looking for, but I can't sell you one because they're all under quarantine from this bug. I was like, oh, what do you mean they're all under quarantine? And then I looked into it. I'm like, I'm gonna fix that. But there's this. Right, so maybe you've heard of a molecule called DDT. It's a problematic molecule, and so this is one of the reasons I've had such a difficult time getting into conservation as a chemist. If you don't know, this is a broad spectrum insecticide that was sprayed all over the world to tamp down mosquito and tick populations. It turns out that it has very limited bioavailability when sprayed in this way. It's basically kind of almost like plastic. But as insects eat it, they die, and then birds will feed on those dead insects, and then it works its way up the food chain and bioaccumulated in raptors and caused the near extinction of bald eagles and peregrine falcons. And so we lost all peregrine falcons in the lower 48 states. And we lost all but, I think it was 200 nesting pairs of bald eagles in America. And if you can imagine America without the bald eagle, I mean, it's just, there's no way, right? And so, through the chemical sleuthing, it was a chemist who made this, but it was a citizen scientist chemist who determined that it was causing the death of bald eagles. And then that set in place regulations. A lot of you may be familiar with Rachel Carson's book, Silent Spring, and I highly recommend reading it. I do not advocate for the broad deployment of chemicals. I'm very aware of this very troubling and destructive situation that happened. And so, as we look to the hemlock forest, this is deeply on our minds. The hemlock forest does get treated with insecticides, but it's done by hand with each tree individually treated. And so here's Dr. Yupu Zhuang, who is a really talented postdoc in my group. He's now building his own research lab at the National Taiwan University in Taipei. And he was really instrumental in helping us understand how we could develop invasive species treatments. Here we're killing some honeysuckle, and he's dabbing a herbicidal chemical on it. But the point is that it's being treated locally. So we're not spraying it at the back of an airplane. It's being applied in a precision setting. And so this is the norm in modern conservation, that we would have a precise mechanical deployment of a medicine. You can imagine, the correlation to medicine would be the surgical removal of a tumor, or the injection of a drug directly into the tumor to try and get it just there. But as a chemist, I've spent a lot of my career thinking about the molecule itself and how we would also have a very selective molecule. And so here, this is actually a broad spectrum herbicide that he's dabbing with great precision onto this tree. And the hemlock woolly adelgid is treated with a broad spectrum insecticide that's hammered into individual hemlock trees. This is the chemical that is used, imidacloprid. It's a neonicotinoid, and you probably have heard neonicotinoids in the news, and it wasn't in a positive context. You might not know that the entire Great Lakes Basin is treated with a chemical, and it has been for 6 decades. So we have an invasive sea lamprey in the Great Lakes. Here's a photo of them. I don't find them particularly attractive, but they completely wiped out sturgeon and brown trout in the Great Lakes. And in the 1950s, it became so problematic that they started investigating ways to kill sea lampreys selectively. And they found both a unique biology to them and a somewhat selective chemical, although it's hard for me in 2026 to look at this chemical and say that would have some type of specificity. It's a simple benzene. It kills lots of fish, but it does kill lampreys more selectively. But there's a trick, is that the sea lampreys only breed up in tiny little streams. So you don't need to fill the big bathtub of the Great Lake. They go to streams upstream of the Great Lakes where the baby sea lampreys are, and they deploy this chemical to the entire Great Lakes Basin, partnership between Canada and America, and we keep the population of sea lamprey in check using TFM. It's largely stewarded out of the USGS facility here in Ann Arbor. It blew my mind when I found this out, right? Because I've been thinking about, like, as a chemist, how could I ever deploy a chemical in any way, and then to learn of the scale that we currently do this. The deployment of TFM to the Great Lakes has brought back the lake sturgeon and brown trout. And so those creatures were nearly extinct, and they are back, and this is one of the tools in the fight against them. But to me, I just look at this and it's too aromatic for me. And you know, one of the things that we think about in modern human medicine is selectivity. And so I show you here 4 cancer drugs used for human medicine. Staurosporine is a highly non-selective chemical. We rarely use this one in cancer therapy today because, as you can see, it's knocking out each node of what's called the kinome. And so this tree that's behind all these pink dots represents the critical biosynthetic infrastructure or biochemical pathways within your body upon which you phosphorylate different things in your body. You've phosphorylated tons of molecules in your body since you sat down here today, and it's because of the kinome that does that. Cancer tends to upregulate a lot of kinases, and so if we attack the kinome, we can tamp down a lot of cancer. And so staurosporine was one of the first chemicals that was found to do this. It's a natural product, and it's nature's solution to stopping phosphorylation. It comes out of, I think it's bacterial in origin, but it's basically a way of a bacteria saying, stop eating me, or you will no longer get to phosphorylate. So we don't use this too much for cancer therapy because it's too broad spectrum. Today, we would use something like selumetinib, which is kind of more like a sniper shot, right? And it's through modern drug discovery tools that we can develop a chemical with such precision, to have such a precise biochemical impact. And you can imagine how artificial intelligence is making this even faster. If you wanted to say, I'm starting from a situation like this where I have no selectivity and I'm steering towards an objective where I have some selectivity, and I'm going to stitch together a bunch of atoms so that carbon has 4 bonds, nitrogen has 3 bonds, oxygen has 2 bonds. Artificial intelligence is a wonderful tool in accelerating these types of discoveries. And so we imagine a future where when trying to kill the hemlock woolly adelgid, we would have both the mechanical precision of approaching individual trees and hammering the drug into them, but we're also very interested in the chemical precision that we would apply if it were a cancer therapy. And so I show you here local boogie-woogie aphids, which are from, this was taken in Scioto Woods. If you haven't been to Scioto Woods, it's a wonderful place to go. It's a little bit cold right now, but in like 4 weeks, it'll be just blooming in trilliums, and I highly recommend you go to Scioto Woods. And in the fall, you might see boogie-woogie aphids there. We were pretty hot on the idea that we could kill hemlock woolly adelgid, which makes that white wax I showed you, by basically stopping its wax production. But then we asked the question, well, what are the local insects that make white wax? And boogie-woogie aphids are one of them. They're just hilarious. You approach them and they dance like this. My daughter hates them. The wax that they produce is the exact same chemical wax that the hemlock woolly adelgid makes. So we're looking for different biochemical pathways, one of which is shown here as this wiggling gray protein. But the fun thing is that using modern artificial intelligence, I can show you this unfortunately pixelated version of a protein of interest. This is coming out of the hemlock woolly adelgid, but we haven't done any crystallography, and we just in the computer can say, take this protein crystal from humans and then now translate it into this invasive insect that we are trying to tackle. And so AI gives us this tool where we can translate findings, knowledge from one species into the species that we want to look at. And with that, we've been able to do so-called docking studies where we can navigate through various chemical structures in hopes that we could find one that selectively targets the invasive insect that we're after, this so-called Adelgis tsugae. And so we found one, this chemical zelandapam is a discontinued hypertension drug. And it turns out it very selectively hits the hemlock woolly adelgids, one of their proteins. It does not impact fruit flies or honeybees or monarch butterflies. And so we're really interested in how we can use modern drug discovery tools rather than looking for selective cancer therapies. Could we try to navigate to a selective insect therapy, but understanding that we're deeply passionate about the forest ecosystem and want to have selectivity for just that one invasive insect. And so, as I hope I've made it clear, it's been difficult to think about what is a chemist's role in this space. I often approach conservationists with some of these ideas, and they're intrigued, but usually I have to discuss why we wouldn't just use genetics to solve the problem, or the microbiome to solve the problem. This tends to be the way that we would solve this. Or actually, one of the main solutions for the hemlock woolly adelgid, the one that I'm always have to answer for why we would need a chemical solution in this space, is for releasing a beetle that eats them in Japan. And so, they have a natural predator in their home environment, so we can bring that predator over and release it here, and we do that. I don't do that, but this is one of the primary control measures that we would have. I guess I would argue is that once you've made that decision, it's very difficult to turn that off, right? And so I talked to you about how DDT caused the near extinction of bald eagles and peregrine falcons. We were able to put in place regulations to stop the deployment of that drug or that insecticide, and the chemical is no longer there. It's no longer a major challenge for bald eagles and peregrine falcons. Whereas cane toads, which were introduced to control beetle pests in Australia, are causing massive ecosystem havoc to Australia because they were a biological solution that didn't go as planned. And so one of the things that I like to say is that chemical tools dilute over time, right? You've heard about PFAS, and so that time that it takes PFAS to dilute is a really, really long time, and that was true of DDT also. But we can understand how to make chemicals that dilute more quickly or degrade more quickly as needed. Whereas biological tools reproduce over time. And so there's a lot of discussion about gene drives in the environment. I hesitate to say I have an opinion on it. I think the science is absolutely incredible, and it's becoming quite possible to have a gene drive that could knock out an invasive species. And I guess what I say is that I think these are complementary tools that a chemical tool at least gives us a little bit more control about how we would discontinue the deployment of that. Okay, so let's spend a moment in the Sonoran Desert with Pebbles the Gila monster, who's actually a local Gila monster. So Pebbles lives here at the Ann Arbor Creature Conservancy. And I'll just quickly close with her story and then move on to any Q&A that might come up. So we met Pebbles. She's at the Creature Conservancy. She's an incredible animal. And unfortunately, she was infected with a cryptosporidium parasite. And so the Wildlife Rescue Center that was housing her had planned euthanasia because they house a lot of really critically endangered reptiles. And so having an infectious reptile in the collection was a no-go. As a pharmacist, I was aware that Ozempic and Wegovy and Zepbound and these modern GLP-1 inhibitors, the very first one came from the Gila monster. And so the saliva of the Gila monster is Byetta. That was the first GLP-1 inhibitor that was marketed, it's Extendin is the Gila monster protein, and then it was marketed as Byetta. And then today we have better products such that DJ Khaled is now selling us this during the Super Bowl, right? So how is it that we're in this situation where there's these multi-billion-dollar pharmaceutical products for weight loss and diabetes that came from this animal, and yet here's a specimen from the species that's about to be euthanized because it's sick. I thought there's got to be something we could do about it. So this was early in our ventures into conservation chemistry. We were struggling to find a way forward, but we wanted to do something, and so we started by getting samples of poop from Pebbles the Gila monster. And from it, we extracted this parasite, which we got an electron micrograph here. And you can see that it's got this really thick black ring around it. And then additionally, this kind of jelly-like coating around it. So we knew that this was going to be a tough challenge. You can't kill this parasite with bleach. So they actually can't even wipe down a surface with bleach to kill this parasite. You can do it with boiling water, but that's not a good therapy. All right, so we got this far. We said, well, we'll spin up in the lab. We'll start making Gila monster cell cultures. I'm not sure where we'll get them from, but we'll find a way to do that, and then we'll develop a parasite infection model, and then we'll screen drugs, and we'll do it the classic way, as though we were getting new tumor tissue from a cancer patient in the hospital. Well, we didn't have time for that, and so the Creature Conservancy said, we got that image in May of 2024, and we said, well, we think we want to make these cell cultures and start doing in vitro experiments. And they said, you're really going to have to do something quicker because the body weight of this animal is just going down, and she's really lethargic, and it's not going to work. So we had another tool that we were using somewhere else, and we deployed it here. So we use artificial intelligence to scrape the literature all the time. And so Tesco Chiganti is a high school student. He was a high school student. He's now doing his undergrad at Johns Hopkins, but an incredibly talented high school student who came up with this tool. So what you do is you use your favorite search engine. We used Google to find a series of papers on this topic. Just quick search, cryptosporidium. What is there? We found 5 papers. So and there are these yellow dots in the center. Now, using tools from the library here at the university, we could say, what's the citation map around those? What are the papers that this paper cites? And you can see that there's some kind of these fireworks of papers that don't get cited by their partners, but there's a lot that, this paper cites this paper, so does this one, and so does this one. So that's probably an important paper to this space. If you take it one step further and say, what do all those papers cite, you get a more complicated map. And so then we get, in basically 2 steps, we can get the entire literature around a topic. And then we used ChatGPT to read the abstracts, to read the supporting information. If the papers were open access, we'd read the whole paper and extract all of the drug names from these papers and get kind of our feet on the ground about this entire medicinal space. And there were 2 molecules that came out of this map of cryptosporidium that were interesting. One was called nitazoxanide, and the other was called piromomycin. And for reasons that I'll happily describe in the Q&A, but in the interest of time, we'll just move forward. We chose piromomycin. So we were going to give Pebbles the Gila monster piromomycin. Before we did that, we treated the parasite in a test tube with piromomycin. We were very happy to see that it had an impact. It was, completely dissolving this dark ring, and there were these white occlusions forming inside of the parasite. We called the Creature Conservancy as soon as we got this image, and within 48 hours rather, they had started her on a course of piromomycin. Formulated in quail. So she got 7 doses of the pill in this format. I'm very happy to report that this graph goes up to the summer, but she tested negative for the disease. She stopped regurgitating her food and since put on all of her body weight. And I got a text yesterday from the Creature Conservancy that she's been in full remission for a year now. So she no longer has this disease and has reached a very healthy body weight. And so we're very happy about how artificial intelligence allowed us to get here. And so Pebbles has been a wonderful friend to the lab, and I can report that the Creature Conservancy has since acquired 2 additional Gila monsters, which they named Ozempic and Wegovy. And so I will pause there. I'd be happy to take any questions, and I just thank you so much for coming on this chilly evening to listen.
Okay, thank you so much. Just put your hand up if you have a question. I will bring you a microphone. I have a question. So in 1951, a guy brings a bonsai tree into his garden, and now we are still dealing with the ramifications of that today. How was it ascertained that it came from that specific bonsai tree in that specific garden? Yeah, genetics. So you can people have been trying to understand this for a long time. They know the tiny Japanese island that the bonsai tree came from through looking at the genetics of the insect, that you can see how it's spread.
If you'll allow me, there's a crazier part to this story, is that in Japan and Asia where the bug is native, it's not a problem over there. It's like I've seen it in Japan and Taiwan, and it's like living in harmony with the forest. It's not a problem. But it's a problem here. But there, it breeds sexually. The bug, lots of bugs do this. They have part of their life cycle is on one tree, and then they sexually reproduce on a different tree. So the boogie woogie aphid that I showed you was feeding on beech trees, and then it sexually produces on larch. The hemlock woolly adelgid is an invader that will feed on our hemlock trees, but it sexually reproduces on tiger tail spruce, which only exist in Asia. So there's no sexual production of new insects here, which means that every insect that has wiped across the landscape since the 1950s is a clone of itself. And so we think that could be like an Achilles heel to this whole problem, right? That like if we can find a way to kill one of them, that it should work for all of them. But it's still a big problem. So
that's crazy.
I think my question sort of is in line with that, because I'm interested in the genetics aspect of it too, because it is a double whammy way to target these things. So can you talk about any exciting things in that frontier, and like looking at the underlying reason of why the cancer cells are metastasizing in the turtles, or what in the hemlock tree is making them susceptible to the invasion, and what in the Gila monster is like susceptible to the disease?
Yeah, sure. I mean, I won't answer those exact questions, but I'll tell you some fun conservation genetics. The vignette I didn't share because of time is the biggest disease event happening on this planet right now is a fungal infection in amphibians. It's called chytrid. It's completely wiped out. You know about chytrid. So I was unfamiliar with it, right? It's the biggest disease event that's happening on this planet, and like I've spent like decades studying disease, and I'd never heard of this. So it just wipes out frogs. It kind of dissolves their skin, and they just can't do. In the Sierra Nevadas of California, there's yellow-legged frogs that the San Diego Zoo is doing everything they can to keep alive. Teams of researchers they only live in 7 ponds. Up in the Sierra Nevadas, and they go and their teams go and treat those 7 ponds with antifungal drugs. But they found that one of the ponds has the population that's there seems to be it doesn't have a problem with it, and so they're trying to learn from those genetics and deploy those frogs to the neighboring ponds in hopes that that population will bring it over. There's an incredible story about synthetic biology, and it kind of on the gene drive-y type space, but I did mention cane toads. And so they're a toad that was introduced to Australia, and they're causing a terrible destruction to the ecosystem there. And they're poisonous. They're like many toads, they're very poisonous. There's a marsupial called a quoll, Quoll. They're absolutely adorable. And they eat these toads and they die. And so they're a critically endangered marsupial because they feed on these invasive toads. They don't know about the poison that's in these toads. So it turns out that the toads came from South America. And the marsupials of South America and the mammals of South America are immune to that toxin. And it's a single base pair edit. So there's like 100 of these quolls left. We know exactly which, and there are CRISPR tools upon which you could switch that out and they would be immune to the poison. Discuss. So that, yeah.
I don't think we have any questions.
How did you decide upon peramomycin?
Yeah, sure.
Yeah.
Yeah, thank you for that question. So it's has a 0% bioavailability, which as a drug hunter, I'd never seen this before. I was like unaware. So what this means is that if you swallow a 1 gram pill, like 1 gram goes through your GI and out into the toilet. Normally we swallow pills so that they get into the blood. And so we were very nervous about treating this animal that it would. It was the Creature Conservancy that treated her, but we were discussing with her vets between these 2 drugs, and we landed on paromomycin because it has a 0% bioavailability, and the disease is in the gut. So we're like, we could target the disease by staying in the gut. And that was the primary selection criteria.
So my question was on the AI use in this system. So my question is like, as a student, when I think of AI, I'm thinking of something I can just use with caution because I don't want to trust all the information I get from it. So in what you talked about, you're talking about how AI is really doing something really important in giving you a good way to work with this synthetic chemistry. So how do you use the answer that you get from the AI to be able to actually do the whole project?
For sure, yeah. Thank you for that question. And I mean, I guess one thing I didn't dig into too much in tonight's lecture is that the different styles of tools that we use. Some of them are AI, some of them are machine learning, and some of them are just straight up statistics. Today, in 2026, people might say, "Oh, it's all AI," but it's just work I did in a computer, really. That's not entirely true. We use large language models, which you might be familiar with, like ChatGPT or these types of things, and those are really prone to this hallucination. That's what I was interpreting within your question. That they'll give you an answer that sounds incredibly confident, and you're like, Then you read and you're like, you made that up, right? Like that wasn't true at all. And so we're very aware of this. That's a problem with LLMs, and that's just one of the tools that we use. What I didn't talk about a lot today is some of the tools that we use to refine signals, not using LLMs at all. We use tools like one that we love to use is called Bayesian optimization. And so Tiger sitting right behind you is an expert in Bayesian optimization. And so we often, I showed you this example where we had this kinase inhibitor that was like pink dots all over the place, and then there was a new kinase inhibitor that just had a much more selective profile. So you can use, we would use AI to try to say, here's my starting point, it's a bit of a mess, here's my objective, and I'm trying to steer in that direction. But we're not using large language models for that. We're not using language. It's all based on like math and numbers, where you can get, you can kind of think about it like if you've made an Excel spreadsheet where you have X versus Y and you make a R squared plot. It's more like that, where you've got like a series of numbers and you're actually navigating through those numbers to find an exact answer. You don't hallucinate so much in that space. But we absolutely use LLMs for some of the other work that we do. It's been very helpful for taking, like I tried to show that we would take information from human medicine and say, like, tell me everything you know about that, but now make it about a sea turtle or make it about a Gila monster. And like it's crazy that you can do it so fast. You need to be really careful that you're not getting fooled by a hallucination.
Okay, let's wrap up and thank Dr. Cernak one more time.
Thank you.